Okay, so my talk is, oops, how do I go back? Unwanted immunogenicity risk in biotherapeutic discovery and development. The outline is basically, I'll talk very briefly about strategies for immunogenicity risk in management and according to our established approaches. Then I want to spend the bulk of my talk on how new tools are being applied to inform risk assessment and mitigation, because this is new science. As with any type of new application of technologies, to a certain extent, our group, which is just beginning to do this type of work, has, to a certain extent, a lot more questions than answers. And I thought it would be a good opportunity to show you some of the questions we're coming up with, and perhaps that could be a framework for some of the later discussions. And then very briefly just talk about next horizon. So I think most people are fairly familiar with the types of events that occur in the immune response. Obviously, there's a lot of focus on secreted antibody, because that's basically what we measure. Is this what I used to point? Oh, this one. Okay.
Yeah, that one should work.
I know what that does. Okay. Secreted antidrug antibody, but we know that there is a number of events that occur earlier, including antigen presentation, T helper cell activation, and engagement with B cells. If there's sufficient signal, then one could get not only activation of B cell and secretion of ADA, but also development of plasma and memory cells, which would be the most concerning. And then there are also these types of events, which we just heard a little bit about, that one could actually get immune tolerance instead, and that would obviously be a desired situation. The factors that are considered, I think most of you have heard a lot about those. Manoj already talked on that. We think about product, patient, and treatment-related factors. More and more, we're also considering other sources of risk, which are competitive and regulatory risk. As I sort of alluded to in some of my questions, in particular to Manoj, we're not just thinking about immunogenicity risk when we first select our candidate. We actually have to think about it throughout the development. Obviously, we want to select the candidates that have the lowest risk. But even in non-clinical development, we have to think about the impact that anti-drug antibody will have on the actual ability to interpret our non-clinical studies. We also want to know which one of the factors, increases of immunogenicity, might actually be applicable to humans, because we usually say it's not predictive in our normal animal models, but we certainly know that there are certain factors, like if the immune response is being driven by drug pharmacology or the target, where our observations may actually be applicable. And then in early clinical development, of course, we want to know anything about pre-existing reactivity. If we see antibody after a single dose, does that really mean that we're going to continue to see increases of antibody, or could we actually see a situation where in later studies with multiple doses we would actually see less immunogenicity? And then, of course, what are the consequences that are likely to occur? In later clinical development and post-approval, we first of all want to know if the immunogenicity that we see is something that can actually be, that we understand it and we know that we can manage it. And then with the change in the manufacturing, dosing regimen, patient population, even the assays, will we see differences in the immunogenicity profile that we have to explain, we have to manage those types of risks. And there are a number of different strategies that we utilize to manage the risk, everything from selection of the study population. In some cases, we might want to select patients with lower risk in earlier studies. In other cases, that's not such a good idea, because then we don't really understand whether that population is at risk or not. And then, you know, the type of testing we do comes into play in the management, as well as timing of sample collection and analysis. And of course, there could be other things, including use of concomitant medications and so on, that can be used to further manage immunogenicity. So we talked before about already talking about things like the reliability of the methods that we're now starting to apply, a lot of these Tcell, in silico assays, et cetera. And I thought it would be useful to go back and, especially since Jeremy says I've been in the business for a long period of time, talk a little bit about what I would consider to be an established predictive immunogenicity risk assessment tool, which is actually our antidrug antibody and NAB assay results. Because it's really not the presence of neutralizing antibodies or antibodies that is the concern. It's really the consequences of those antibodies. So a positive result could be considered a risk factor for the consequences, right? And if you think about how we utilize those types of data, there are routine assays that we now run all the time as laboratory assessments. But going back to my early days, I can remember times when we saw this type of data, we were presenting these types of data, and it doesn't necessarily always correlate, oftentimes doesn't correlate with clinical effects, and certainly we got similar types of questions. Well, why are we doing these types of measurements? They don't seem to correlate with anything. What does a certain titer tell us about the risk for that patient losing efficacy or losing or having hypersensitivity reactions? And our answers were usually, we don't know how titers correlate with clinical effects. So we talk about validating these types of assays, but preclinical validation is really done with surrogate antibodies. We measure things like precision and sensitivity, but we really don't, if you're really experienced with these types of assays, you know that surrogate antibody doesn't necessarily represent even the type of performance in the assay that you would get from actual patient responses. When we talk about clinical validation, which is what we're kind of being asked for these new tools, that really requires a significant amount of compound-specific data. And you wouldn't necessarily say, well, we showed the correlation of an antibody titer assay for compound A, and that therefore we're going to have good correlation with any antidrug antibody assay that we do with clinical effect in other indications. So I think it's important to recognize that. And we certainly have a lot of false positives, especially more recently as these assays have become much more sensitive. We oftentimes see antibodies that have no effect, nor where the titers correlate with effect. And we know that we have interferences that can prevent you from detecting antibodies, and true positives are often clinically irrelevant. On the other hand, we also see frequent false negatives, especially with drugs that have a long half-life. We may not see antibodies because of drug interference. Bridge assays don't always detect all of the immunoglobulins that could be present. We may not see low affinity antibodies, not all the epitopes are recognizable. And sometimes when we have false negatives, we really do have an effect. We use this tiered approach because these are, while not as expensive as T-cell assays, it's still expensive to do these types of assays if we did a neutralizing antibody assay for absolutely every single sample. So we use a tiered approach, and we recognize that by using that approach we may be missing some things that are informative. But despite all of these limitations, we're very used to doing these types of assays. After a few decades of doing it, we understand the limitations, and we do use it for monitoring and making decisions. So if, on the other hand, we start looking at some of the emerging predictive immunogenicity assessment tools, like the T-cell epitope assays, you can see a very similar type of pattern. It's possible to do a tiered approach, starting off with the less expensive, easier throughput assays like the in silico analysis, going to your in vitro binding, your ex vivo assays, and then things like in vivo assays. Clinical trials are also a place where we're trying to predict, again, looking at whether or not we see a response in a phase 1 study that could tell us what will happen when we expand to a much larger population or a different type of population. And the types of, the rationale for using these assays is that T-cell activation is a fairly key risk factor, and I don't think there's a lot of argument about that. It drives higher affinity anti-drug antibody responses, persistent responses, memory responses, type 1, 3, 4 hypersensitivity, and so on. And so, you know, I oftentimes have a trouble when people say how relevant are these types of assays, because we know that T cells should be involved. We know that, you know, just as with an ADA, clinical validation is something that would have to come over time, and it's probably compound specific. The nature of the epitope is much more predictive for T cells than B cells. We do have advanced tools available, and you know, if we choose to do so, although this is pretty cost prohibitive, we could use the later application to actually try to get some clinical validation. The questions we ask are there sequences that actually look like T cell epitopes? Are they actually germline or unique sequences? What coverage do we have in the population? If we see T cell epitopes, are they just going to be covering a small part of our patient population, and so on? And then, of course, we would do confirmatory assays to ask additional questions and probe that more. The questions like, are there actually responsive human T cells present? Are these sequences actually capable of driving an ADA response? And then, again, if we chose to, we could probe further in clinical studies. So I think, you know, probably most of you understand the concepts between T cell assays. In a lot of cases, we're looking at T cell proliferation or activation as a readout. But things like the in silico preparations and the in vitro binding really focus on the MHC class II binding properties. We have to consider that things could be happening in terms of the actual immune responses, because just because something binds to HLA, we know that things like inflammation and activation could actually enhance the amount of HLA expressed, and so on. Proteins need to be actually transported and processed, et cetera. And so not all of the assays measure all of these types of analyses, but some of them do. So starting with the in silico T cell analysis, again, there's an advantage here. I know that there's a lot of skepticism about in silico, but really I think this is a place where we all want things to get better and more reliability, because it's really nice to be able to just take your sequence, your theoretical sequence, and put it into a program and get some kind of information about the immunogenicity risk. And certainly throughout biology, there's an emphasis on trying to do more computational and modeling analysis. So, you know, this is an area where as an industry it seems important to work together. And you can answer a lot of these questions from a lot of the programs. So once you actually invest in the technology, it then becomes less expensive to do it. But it doesn't address things like processing at this point or other requirements for immune dominance. There is some evidence that it works in some cases. As others have said, there are certainly examples where it hasn't been predictive, but this is probably one of the classical examples, the Amgen study, in which they showed that the peptide that they showed as being more problematic by in silico analysis did turn out to be the peptide, and you know, the HLA types as well did turn out to be the HLA types and sequence that seem to be much more responsive in recall responses. So that gives some kind of confidence, although those types of examples, those clear examples, are still not that many. So I think where a lot of the use of this type of tool is in looking at, especially as a lot of our companies and organizations are trying to make better sequences, molecules, and so obviously when our molecular designers are trying to determine what to actually use as replacements, this is a very nice tool to be able to look at, you know, various amino acids that they can actually use both in designing CDRs or in designing mutated sequence when they're trying to make a better. So here's one of the first questions we have is, So it's going to be fairly simple if you have a situation where you made this mutation and the mutation actually decreases the signal that you see in this type of assay. Or vice versa, if you propose a mutation and it actually apparently increases risk, that's fairly comforting as well. But what we actually see a lot is where we see the mutation being made in a sequence which actually is already considered to be a T-cell epitope. And when you introduce the mutation, it doesn't really change the signal that much. We still see T-cell epitopes. But the question is, have we actually increased the risk? If this is something that this is a region that can actually be presented on HLA, and in the wild type, it's germline or however you want to describe that. But now we've made a mutation. Have we actually increased the immunogenicity risk by doing that, even though we haven't increased the HLA binding? So I'd be very interested in discussions on that particular point. Then when we talk about assessing the validity of this, I don't prefer to think about assessing validity in terms of, well, did that really translate to an immune response? Because, first of all, it will be a long time before we get the immune response. And whether or not we get an immune response is going to depend on a host of multiple factors, besides just whether or not there's a new epitope that we've introduced. But the in silico tool is really supposed to be predictive of HLA binding to particular HLA types. And so I think that that's really one of the areas where we need to think about, does it predict what it's supposed to predict, which is HLA binding? And so Tim Hickling in my group, and I think he got this format off of somebody else who had presented in another meeting, was looking at some of the data that we've started to accumulate. And as I said, we're relatively new in this field, so our data sets still are pretty low. But in looking at those type of data sets, you can compare things like the true and false positives that you get both from in vitro and in silico analysis, and try to get a sense of how predictive it is. And in fact, I think this is going to depend on the cutoffs for how many of these true positives actually, how many of the positives from in silico actually do turn out to be true positives. So we actually did expect to have a relatively high false positive rate, because the expectation was that then you would do the in vitro analysis to determine whether it was really a true positive. But the bigger concern is really whether there's a high false positive rate. And I think both in our limited data set and the data set that was presented by this other person from this other organization, in both cases, which was notable, was that it seemed to have a high false negative rate. And so that could be of concern. And so here's an opportunity perhaps where cross-industry we could, even if we don't share like what the sequences were that were generating these type of data, there's an opportunity to present data in a way that is fairly anonymized, but with similar types of formats, so that we can get a better idea of the reliability of these tools. So, it's an opportunity to do that type of thing, especially for companies that are doing both the in silico analysis as well as the in vitro binding assays, and more recently DCMS assays are coming into favor. So, based on that previous data set, we came up to the conclusion, well, it's got some level of reliability, probably pretty good for making some of those really early decisions where we have nothing else to go on. But here's a more significant concern, I think, which is if you're just looking at if it's positive or negative in either one of those methods, there seems to be, maybe a reasonable correlation. But, affinity and stability is supposed to be an important factor, because that's supposed to determine whether or not that peptide stays on HLA and whether it's going to be on that HLA molecule for a significant enough period of time so that the T cells that could recognize it can come along and actually be able to find it. And so if it's not very stable, then that could decrease your chance of actually inducing an immune response. That's where I think these types of tools are showing, even though there is a correlation, it's not actually very good. And so that's certainly an area where there could be some improvement in terms of that. I just wanted to present this other piece because, you know, when you think about how these correlate with the risk of other types of risk factors, we worked with one of our colleagues, a couple of our colleagues in our PharmSci group who are interested in the risk of aggregation. And it turns out that there are in silico tools that can predict aggregation propensity. And again, you know, those are in silico tools. They have a certain level of reliability, and they're very specific for certain types of aggregation-prone regions. And one of the things that they found was that these aggregation-prone regions, in particular these beta crosslink regions, seem to correlate pretty well with Tcell epitopes as predicted by in silico analysis. And one of the things we're trying to, you know, decide within our own organization, and this has all been published, is what does this actually indicate to us? Is there something in common between aggregation-prone regions and T-cell epitopes? Is it a coincidence that, you know, maybe they're just hydrophobic regions, or is there some other reason that we're seeing this correlation? Again, I'm just presenting this as an area that we'd like to understand more about in terms of you know, why are we seeing these types of coincidences? Is it really telling us something about what the immune system is looking for when it's actually scanning for T-cell epitopes and things that it wants to present to the immune system? So obviously moving along beyond the in silico analysis and the in vitro analysis, PBMC assays are available to help us answer other types of questions, including whether there are T cells present that can actually tell us that we have an immune response. There's a number of different readouts for these types of analyses, including measurements of proliferation, cytokine production, activation markers, and so on. So we, again, have just started to utilize these types of methods. There are multiple parameters we found when we look at the data. One of the questions is, of course, how do you actually present the data? This is a pro-immune assay, and the reports we get show the data presented in a number of different ways, which is very nice for doing the analysis. Again, our analysis was looking at a mutant versus a wild type, and you can look at the number of donors that actually do respond. You can also get data that shows the responses according to the magnitude of the responses. And then there's a combination analysis that shows you not only the number of donors that gives you an index that's related to both the number of donors that respond as well as the magnitude of the responses that we're seeing. And you can see that in some cases, let me see if I can go back, you know, it may look like we have a number of donors that are responding, but the responses can be very heavily skewed by a few donors, some of whom may respond very well to multiple antigens. So as we start to look at these data, one of the first questions come up is what's the appropriate cutoff? Again, we get reports that have a number of different analyses using different cutoffs, both for the index as well as the standard error. And as with all assays, similarly to what we see with anti-drug antibody assays, when you look at your results and you say, how am I going to use those for ranking, one of the concerns is that we might rank them differently depending on what the cutoff is. And so again, I think this is an area where we can have a number of discussions about how individual groups are setting their cutoffs and what over time we're going to find to be the most meaningful cutoffs. Another thing we've seen for this particular analysis, again comparing the mutant and wild type, and this is a monoclonal antibody in which there were some modifications made in the antibody, and we wanted to look at both the full region in which those modifications were made, as well as some overlapping peptides from within the region. And our expectation was that the overlapping peptides would have similar responses, but would represent a subset of the responses that we saw from the full-length peptide. And in fact, we didn't actually see those results. What we saw was that some donors responded to the full-length peptide, and we saw a different set of donors responding to the smaller peptides. And you know, that brings up the question of, does the size of the peptide actually, or some of the properties of the peptide actually affect how it's actually taken up, and makes it more complicated for deciding how to actually tier your analysis so that you can narrow down what are the problematic regions. So very interested to hear if others have seen similar type of effects. And then, of course, the DC T-cell assays are now popularly being used. They can answer other questions, and in particular, they're useful in situations in which you're looking at products that are actually immunomodulatory, because you can see what the effect is when you actually load the protein into the DC assay a little bit separately from putting it into a mix of all of the immune cells. And I'm not going to go into detail about that, how it's actually worked, because that's already been covered. But what we wanted to do was look at a number of monoclonal antibodies. We actually had, for our initial analysis, we were looking at one antibody which actually had immunogenicity. We had some clinical immunogenicity, and we wanted to look at some backup molecules. In addition, we wanted to compare this with another molecule which had a low level of clinical immunogenicity. These three molecules are related in that they all have a similar target. This one has a different target, but we did want to look at two molecules where we had some clinical immunogenicity data. And again, looking at things like the in silico analysis, what you see is if you just look at total number of T-cell epitopes across the whole molecule, I think what you'll find is that immunoglobulins, because they're a very large molecule, have quite a few cell sequences that will show up as T-cell epitopes, and it's sort of hard to distinguish by these Z-scores alone what's going to be more immunogenic than others. If you start looking more carefully at the actual regions that we think would be more problematic, which would be the CDRs, any regions in the FC that might be mutated, and so on, it's going to be more informative. And so we looked at these molecules and also compared it to adalimumab, which was another molecule where there's clinical immunogenicity data. And using the EpiVax tool, you can actually get information that tells you which ones of these, where there are epitopes. We can also look at where those epitopes are actually in CDRs, where they're actually showing up in germline sequences, which we predict or we expect would be less problematic from an immunogenicity perspective. And then, of course, EpiVax has this other tool that can tell you whether or not there are sequences that they believe actually activate T regulatory cells or T regitopes. And so this is sort of the way we look at the data in order to try to look at the most problematic areas. And so based on this, these were the T regitope adjusted scores. So one of the important things is most of these are in the negative numbers, so they're all falling into the area that are considered to be less immunogenic monoclonal antibodies according to the way that EpiVax presents this. But nonetheless, some slight differences among the different proteins. So then, looking at the data from the DC T-cell assay, a couple of things that we note is that, the two molecules where we have clinical data, we do see a difference. I guess it's reassuring to say that they score differently, and using various cutoffs, we continue to see those types of rankings scoring, consistently. So it wasn't like in this case that we saw them ranking differently depending on how we actually analyzed the data. But of note, we only looked at one concentration, which ProImmune has said they have optimized. But we still have some concerns about that because, in particular, when we look at the difference between this one molecule and the other two that we're interested in as backups, the question is, how much of a difference do we need to see to say, okay, so these molecules here look like they're lower risk? So again, a potential point of discussion. Another thing that we note is that there are going to be other factors that, even though these two, the high and low immunogenicity proteins scored fairly similarly in the in silico, we do see differences both here and in the clinic. And some of the things that can contribute is the germline T regitope content, because they are different between the two molecules. The target cells are different between the two molecules. And of course, the in silico doesn't tell us about the efficiency of uptake and processing, which the DC T-cell assay does incorporate. However, we don't know which of these factors from this type of analysis is really playing the role. So, one of the questions we have is whether or not we could, if we looked at multiple concentrations here, could you see more distinction between these? Because I think in a lot of biology, it's fairly typical to try to differentiate based on suboptimal conditions. So, that's one of the other questions we have is, would suboptimal conditions as well as optimized conditions give us a broader picture? And actually, Li Zhu in our group is looking at that, and we'll probably be presenting that at some later point. So next, horizons. I think our approach was to sort of start off with the tools that were already available, where there were companies or organizations that already had well-defined processes and established methodology, so that we could start to utilize that and begin to make decisions and understand how we could utilize those to de-risk our portfolio. But I think most of us feel like they have some value in assessing particular risk factors, but there are multiple risk factors that need to be incorporated, and then there's the question of how these all work together. So really what we see on the new horizon is we want to explore more integrated methodologies, things like the artificial lymph node, humanized mice. There's also a laboratory that's looking at a reductionist approach, where one could look at things like the actual processing by proteolytic peptides. These could all have value in different ways. The reductionist approach could obviously be valuable in terms of determining what the reason is for the particular effects that we're seeing. And that's going to be important in terms of developing mitigation strategies related to molecular design. The integrated models will give us more information about how these types of risk factors, or what happens when multiple risk factors come together. And then, of course, it's already been mentioned that immune response modeling is an area that's coming forward. I think as we begin to get more information from all of these tools, that's going to be an important thing to put into play, and there's already some efforts in that area. So I originally had conclusions down here, and at Tim's suggestion I changed it to discussion points, because we really don't have a lot of conclusions at this point. The question that we're left with was, does application of these new tools offer opportunities to improve risk assessment? We think yes, but with a strong but. As with any new technology applications, right now the questions we come up with when we see this type of data overshadow what the conclusions that we're making. And as I already mentioned, you know, that whole concept of how do we actually link it to clinical relevance is going to be challenging, but I don't think it's any more challenging than it was for when we first started getting ADA a few decades ago. Similar types of questions are going to come up. What do we use for cutoffs, right? So that was the case for ADA a long time ago. Everybody was using different cutoffs. Do you want a cutoff that's clinically meaningful, that correlates with clinical effect, or do you want to see everything that's there that could possibly be a risk so that you could then monitor and mitigate based on that? Sensitivity will be a question, but the question is sensitivity for what? You know, is it going to be sensitivity in terms of what it's specifically trying to predict when we talk about things like in silico in vitro correlation, or and similar type of question for validation. What are we trying to validate? Are we trying to clinically validate in terms of clinically meaningful immune response, or are we just trying to validate that tool for what it's supposed to predict in terms of the type of risk? Lots of opportunities for collaboration in that area. I would say that one of the first areas could be, can we come to agreement on how to actually frame the questions? What are the questions we're trying to ask with these particular tools? Can we come to some agreement on how to design protocols for utilizing these tools? How we would decide what controls to use? Do we want to be using controls like PPD and KLH? Or do we want to be using controls that are monoclonal antibodies or biotherapeutics and so on? And that might drive the sensitivity of these methods and so on. And then I'm sure there's more others that we could agree on collaboration for. So I just want to acknowledge a few of the colleagues that I've already mentioned, Tim and Lee, who are both here. Chris Lepsey is our discipline sponsor, who's been very helpful in trying to help us navigate that whole area of how we actually make a business case, even though there are still a lot of questions. I think one of the nice things about being in PDM is that a lot of these same sort of questions were asked a number of years ago by some of our leaders, or too many of our leaders, in terms of whether one can predict things like metabolism of small molecules and pharmacokinetics of small molecules. And you know, most of them agree that there was equally as much skepticism, and it was only with a lot of industry collaboration that some pretty impressive in silico tools have been developed to do that type of thing. I already mentioned that we have extensive collaborations with other colleagues within Pfizer who are involved in mitigating risk, identifying and mitigating risk. Showed some data from the PharmSci, and of course we've been doing a lot of work with ProImmune and have had very helpful discussions with them. So thank you.